Good morning, everybody. Thanks for waiting a couple of minutes for us to get through some technical issues, but we are here now and we're excited to chat about our BeCrop 3.5 updates. To introduce ourselves really quickly before we hop in, looks like we've got 77 folks here today. I'm Marie Mullins. I'm one of our account managers at Biome Makers. I work with our clients across the US, primarily in the Western US, to help understand and utilize our technology in the marketplace. Anything from growers and using our technology to make field diagnosis through ag input manufacturers and using our trials to validate product performance, and retailers and dealers in between. So excited to be here, and I'll let Gus introduce himself really quick, and then I'll go over some housekeeping items before we dive in.
Yeah, thanks, Marie. I'm Gus Bauman. I'm a technical agronomist with Biome Makers, and my role as an agronomist is mainly focused on education, understanding of our soil biology data. So just ensuring that our clients across North America maximize the agronomic value that they can generate from our BeCrop soil biology data, turn it into actionable insights that ultimately support regenerative ag practices and soil health focused management to help ensure that our soils are more resilient, productive, and able to support growers' bottom line. So really excited to talk through some of our updates with our BeCrop 3.5 iteration today. Thanks you all for joining.
Thanks, Gus. So before we dive into all of the really awesome content that we have prepared for you all today, this webinar is being recorded, so just keep that in mind. It will be up on our website in the next day or two following this presentation. And we love participation, we love questions. We'll have plenty of times for questions at the end, I believe. So if you have any questions, please drop them in the chat and we will answer them and go through them. If we don't get to your question, don't worry. We saved them all and we will find you and get an answer back to you in the next couple of days. So with that, I will jump in. Before we get to the first slide here, I just want to kind of frame up why we're all talking about this today and where these 3.5 updates came from in both our BeCrop reports and our BeCrop trials. So, the industry as a whole has been having all of these questions around how do we use biological products? How do we know where to put them, where they're going to make the most impact? And so there's these, a handful of biological testing, organizations like Biome Makers that all have kind of had the same question. Like, we have this information on the functionality of the microbiome. What do we do with it? How do we make actionable decisions out of this data? The biology is really complex. It's a complex ecosystem. The soil microbes in different, on a different field side by side are different, let alone across the country in different cropping systems. So there's a lot of variability. There's a lot of diversity in the soil ecosystem. So how do we decode the soil DNA and turn it into action items that we can use to position products, to understand our management practices and so on at the field level? So that's where the 3.5, next step in our technology is born from, is trying to take the soil intelligence data into actionable information on the field. So we're going to talk about our BeCrop reports, our agronomy reports or field level reports today. And then we're also going to talk about our BeCrop trials. And within both of those, we've made updates to have more go-to-market information, actionable information on them, so they're easier to understand and easier to digest and translate the data from the field or from the trials directly into actionable insights for agronomists, retailers, growers, and ag input manufacturers. So just kind of wanted to tee things up there and talk about how we got where we are and why we're talking about this today. And it's a really exciting evolution for us. And so we're really excited to share it with everybody. So again, like I mentioned, today we're going to kind of just, for those of you who are new to Biome Makers, we're going to do a little quick overview of the BeCrop technology and what we do and why we do it. And then we're going to dive into our BeCrop tests first and go over, what our BeCrop test is, our new updates to the BeCrop test, and we're going to go through a few examples of how we can make actionable decisions and around biological products. And we'll talk a little bit about the biological product landscape and how busy it is and why having this information will really help to inform better decision making on the farm. And then we'll also talk about our BeCrop Trials. So our trials, we'll go into details later, but we're going to go over our new cover page for the trial that is ready to take to the marketing and sales team to educate the team on the mode of action of products, and talk about our biosustainability metric that we've integrated into our trial reports as well. And I think we're going to have plenty of time for questions. So excited to dive in and get some feedback from you all as we go. So for those of you who are new to Biome Makers or needing a refresh on what we really do, we're going to just have a quick whistle-stop tour of what our technology is before we dive into our different product offerings and services. So at Biome Makers, we have our BeCrop technology, and what we're doing is extracting DNA from the soil, turning it into biodata. So our raw taxonomic information on bacteria and fungi, and then we take that and put it through a predictive tool, our proprietary AI and computer learning, and turn it into these functional reports that maps the diverse biology and all of the bacteria and fungi that we identify in the sequencing into these functional reports that are now made very actionable. So we're looking at things, we'll dive into that, but looking at things like nutrient cycling, disease risk, all of your plant promoting growth hormones and stress adaptation hormones, and our micronutrients. So, again, just like your chemical fertility analysis that you take every year in order to, diagnose and prescribe fertility plans for the year, we extract DNA just like you might extract chemistry. Different methodology in the lab, obviously, but we are extracting the DNA and turning it into reports that are designed to make decisions around biological products, just like your fertility analysis helps you make decisions on your fertility and your fertilizer for the year. Also the disease pressure side of things, right? So that's not something that comes with your fertility analysis, but you can make more informed decisions to improve ROI around disease. Maybe some fields have higher pathogen risk than others, and we can use that in our decision making on where to more precision apply, biocontrol or different synthetic pesticide or fungicide applications. So that's really high level what our technology does. And just to kind of go through again, I mentioned our proprietary tech is taking the raw taxonomy reads from the DNA sequencer, which and taking that and turning it into the functional report, which uses artificial intelligence, computer learning, and deep ecological modeling to look at the whole soil ecosystem. We have over 17 million microbes in our database, and this 14 million, at the end of this year, we'll have over 17 million, so we've grown, and every year we're adding millions and millions of microbes to our database. We have a soil database that has soil samples from across the world, and we have over 170 crops in our database. So we have the largest global database to date in terms of soil biology, diversity and cropping systems, and a global database that covers over 45 countries. So, that's kind of the robustness of our intelligence on the back end of our system. So all of these updates that we're going to talk about today go beyond just linear a linear decision-making pathway where if you have, like you would for fertility, right, where if you have low nitrogen, then you add nitrogen. Our database is, and our algorithm is using intelligence from hundreds of thousands of soil samples across many countries, across 170 cropping systems. We've looked at almost 400 different biological products. So we're using all of that insight to deliver these suggestions and start to dig into more actionable items on our reports. So it's really exciting stuff that we're going to dive into. And so just to recap the data that's provided in our BeCrop reports, and this is true for our BeCrop report as well as our BeCrop trials, where we're looking at all these different layers of data. So first here we have our soil quality metrics. This is where the ecosystem functionality really comes in, looking at how the whole ecosystem is working together. We're looking at biodiversity, which is pretty straightforward. How many different types of species are there? And then we look at their overall functionality. So how well are they playing together in the ecosystem? Are they interacting and promoting each other, or are they competing with each other? So what our technology can do, which is unique to ours, really is unique to the type of sequencing and the modeling that we do, is map the whole ecology, right? So that's why we're adding millions of microbes a year, and we're looking at how they're all interfacing and working together. So we'll dive into that more later, but that's kind of the soil quality index, is looking at the ecological network and ecological health and functionality of the whole soil microbiome in that field. And then we have our plant health metrics. So these are really important, right? We have our disease risk. So I think we have over 2,000 diseases in our database. Some of those might overlap if they are impacting multiple crops, but we have a different disease panel for every crop in our database. We have over 170 crops. And so we're looking at disease specific to that cropping type, and we're analyzing disease risk overall. And then we're looking at what natural biocontrol agents you have present in the field. What do you have to promote disease resilience? We're also looking at phytohormone producers, so your auxin, cytokinin, gibberellin, and then other stress adaptive metabolites like exopolysaccharides and siderophore. We're looking at the potential to promote plant health on multiple levels. And then lastly, we're looking at nutrient cycling, which is obviously really important. And all of our nutrients in the soil are largely driven by biochemistry. They're made available through biochemical reactions. So understanding what biology is there to move nitrogen, phosphorus, potassium, all of those natural cycling pathways to increase our ROI on our fertility, our carbon pathways to understand the overall carbon cycle in the soil biology, organic matter breakdown, and our micronutrient pathways. So we have 8 micronutrients on our reports as well, which we'll talk about in a bit. But we're really covering the whole plant ecosystem of hormones, disease, and nutrients, and all of the things that are really critical to having a thriving crop and understanding the whole system. I'll mention here too at the bottom, oh, sorry, Gus, that was my bad. I'll just add really quickly our fertility analysis here at the bottom. We have a few different partner labs. Region Ag Labs is one of them. They're now running the BeCrop test in-house, which is really exciting. They're in Nebraska, and they run our Haney soil test, or their Haney soil test, and we can add it to your reports. And then we also have Malic 1 and Malic 3 and total soil digestion. So with any of your biology tests, whether you send it to one of our partner labs or directly to us, we can have both the biological analysis and the fertility analysis done together, which when you put those together, you really optimize your decision making, right? Having what biochemistry is going on in the soil plus the chemistry itself can inform better decisions in the field. So, moving on here, why does all of this matter? What do we do with this information, right? Largely we have this biological input landscape, which I don't know if everyone has seen this diagram. It's really busy. There's so many labels. There's so many types of products. There's just a lot going on in the biological marketplace. And one of the reasons that Biome Makers was founded was to take the noise out of this, to fill in the black box that helps agronomists, growers, ag input manufacturers understand a couple of things. One, what their product is actually doing and how it's influencing the soil biology based on the cropping system. And then, I don't know, if there's a lot of you here that are agronomists or growers, I don't know if anyone's experienced this, but I hear this a lot. You have you use a biological product in 2 fields side by side, and in one field it has tremendous impact and you are increasing your yield by 5 to 20%, and then in the other field it does nothing. Why does that happen is what we're really trying to drill down into, is understanding the baseline biology, so that when we put a biological product down, we're doing it with the insight that will make sure that we're putting the right product down that's actually needed, that's actually going to fill a biological bottleneck, as Gus says. Like, how do we make sure we're addressing the issue that's most needed in this field in order to promote yield and increase production and ROI? So that's really what we're getting to here, is how do we make decisions on biological products? How do we weed through this really busy biological landscape? And what we're going to talk about today is really, we can do that by understanding our baseline biology and what's going on, and knowing what are the top 3 things, the lowest hanging fruit, that are going to really move the needle for us in a biological way, and increase nutrient use efficiencies, decrease disease pressure, increase hormone production. What are those things that your field needs the most, this field needs the most, and how do I look into a product portfolio, whether I'm a grower looking at my agronomist portfolio, or I'm an agronomist and I'm talking to my grower about what they need? How do I look into a portfolio and decide which product is going to actually move the needle? So that's kind of how we got to where we are, and why we're really excited about these updates is because this is what everyone's been asking for, is the so what. We have the biology data, how do we link it with this really complex marketplace? And so that's what we'll be diving in here. So first, I just want to visit the biological landscape one step further. So let's talk about what different types of products there are. There are 3 primary classes or groups of biological products. We have biostimulants, biocontrols, and biofertility. Biostimulants are the most common that we see. And, so biostimulants can be very specific all the way to very broad. They're in general promoting microbial activity, and depending on the biostimulant, they have different modes of action or different ways of doing that. Whether you have a carbon-based product or an algae-based product, they have different ways of promoting the biology. But in general, they're all trying to promote biological activity without bringing new microbes in. They're supporting crop resilience and stress tolerance. So a lot of biostimulants that we look at are really moving the needle in terms of hormone production and stress adaptation, and sometimes nutrient cycling. But all through the mechanism of promoting biological activity. So then we have biocontrol agents, which, there's a lot of biofungicides out there, but they're really designed for disease treatment. And sometimes they either contain actual microbes that are naturally predatory to the pathogen, or they're natural compounds like terpenes that will suppress the pathogens. And then lastly, we have biofertility products. So biofertility products are those products that are living inoculants. You're actually bringing new biology in, and your goal is to increase nutrient uptake and nutrient availability. Sometimes they're organic fertilizer products or different types of biofertility products, but we're usually bringing something new into the ecosystem. And those things are meant, again, to promote your macronutrient and micronutrient pathways and availability. So I wanted to frame this up because it's really important to understand to just kind of identify the primary classes of products, because when we look at our BeCrop report, it's set up just like this. It's set up to address your hormone production and biostimulant related things. It's designed to address your biocontrol, your disease related things, and it's addressed to look at your biofertility and nutrient related things. So we're going to talk about that, but I just kind of want to frame up. We have 3 primary sections of our report that'll speak to either biostimulant, biocontrol, or biofertility, which makes it really straightforward to take that information into action in terms of just selecting which products are needed in that field. So before we dive into the reports themselves, part of these updates is also having an ability to look at specific outcomes or goals within the projects that you're working on. So whether that's at the field level or the farm level. So we have different project categories. So if you're trying to identify, and based on what project category you select, which is optional, but is something that you can do to have more specific reports, is yield, which will help identify yield limiting bottlenecks, like nutrient mobilization is usually a really big one. And then we have BeCrop management, which helps evaluate management practices and differentiate fields based on various management practices and look at them side by side to look at how management practices are influencing all of these biological indexes. And then lastly, we have disease risk and looking at disease. So how do we prioritize biocontrol, disease risk, promoting plant health to limit disease risk? So I just wanted to, these are kind of the new project types you can select if you want to get more specific insight on different categories.
Yeah, and one quick thing just to add to that. So as Marie said, with these project types, talking to our commercial team, you can select these 3 based on, of course, your objectives. But the focus here, it mentions for BeCrop yield, the prioritization of nutrient cycling. For BeCrop management, that's looking more broadly at management practices and programs and evaluating their effectiveness in terms of soil biology. So that's more of a balanced approach to looking at disease and nutrient cycling both as major factors. And then BeCrop diseases, that those projects focusing primarily on disease, of course. So disease risk levels and biocontrol are the main metrics highlighted. And the reason I just wanted to point that out is because that's what's focused on the new cover page that we'll go over in a minute. So this project focus allowing the individual BeCrop reports to be tailored a little bit more towards the goal of larger sampling projects.
Thanks, Gus. So now we're going to dive into the updates here. And again, just to kind of recap, what were the goals for this new update that we're releasing, is to really simplify the process for identifying biological deficiencies and creating more ease and efficiency around placing agronomic solutions like biostimulant products or other biologicals using the BeCrop test results. So the update is prioritizing deficient biological functions that are the primary drivers in plant yield. So, again, we have our primary functions, which are the most impactful to yield, things like disease risk, NPK cycling, fungicide agents, which help balance disease risk. And then we have our secondary functions, which are plant growth promoters and nutrient cycling. So we'll look here on our new reports, but what we're the new reports, which Gus will go over here shortly, it has a front page that is just going to identify the biggest hotspots. And so you could just look at that and make your decisions from there and not have to go, you'll still have the full report to go through with all of the nitty-gritty details, but you can just focus on, okay, I have a lot of fields here. I just need to know what's the biggest problem areas that's going to help move the needle in terms of yield the most. I'm going to address those first. And you can just have easier decision making based on that one first page summary and insights.
And one thing I just want to spend just a quick minute discussing here with the update, the prioritization on that first cover page of biological functions that we consider the primary drivers of yield. I already know and anticipate we'll get questions about, well, how do you determine what the primary drivers of yield are? And of course, there's of course variability across crop type in terms of what the main drivers are, but in general, based on decades of agronomic science, largely regarded as kind of the baseline or the foundation of crop health are these 3 primary functions: disease, our macronutrients, which most crops, not all, but most crops need in much larger quantities than most of your micronutrients. And then fungicide agents, those beneficial microbes that are able to suppress more of the problematic pathogens, which happen to be fungi. And I think the best way to kind of think about this is from the perspective of the plant health pyramid, if any of you are familiar, which Advancing Eco-Ag and many other regenerative ag consultants and advocates focus on. And not to go into too much detail on it, but basically we can think of it like the foundational needs of the crop in order for it to establish enough productivity to ultimately pan out in terms of yield. These sort of basic needs should be met. A crop really, it's going to have, a field is really going to struggle if disease is out of control in terms of prevalence. So any high risk diseases where we're seeing high quantities of that pathogen, fungal or bacterial in the soil, that will pop out on the first page. And then of course NP and K cycling are kind of the backbone of, like I said, needed in much higher quantities than your micronutrients, and then fungicide agents helping ensure that diseases don't become problematic throughout the season. And kind of like, in terms of humans, we talk about our hierarchy of needs being, needing at least, food, water, shelter, our basics in order to achieve, higher levels of satisfying our human needs like social interaction and hobbies and such. So these are kind of those for a plant. When we think about the plant health pyramid, the basis of it is being able to achieve strong photosynthesis and then protein synthesis. In order for plants to reach that sort of base level of their need, these primary functions we know agronomically are the main drivers. So just wanted to tackle that topic really quickly. And this sort of focus on these primary functions, and then if none of these primary functions are problematic, that's where the cover page will look at some of these secondary functions if they're problematic. So again, it's based on which metrics are deficient or optimal in terms of their BeCrop score ratings. So Marie, I can take it from here on the updates as far as the cover page, but of course, feel free to jump in with any comments if I miss anything. So here on the right, we have the example of this new cover page, just a demo report. And like we've mentioned, what we have on this page is highlighting the 3 top metrics for improvement. So we list out, based on those metrics that we know to be most impactful on yield and crop productivity, if our algorithm will prioritize the ones that, if they are deficient, or in the case of diseases, if they are very high prevalence. So in this particular report, we can see charcoal rot was detected at the pathogen responsible for charcoal rot was detected at very high levels, whereas our nitrogen and potassium pathways, the levels of microbes that cycle and unlock nitrogen and potassium, were very low and low. So those being, the 3 big interest areas to potentially improve the health of this and productivity of this field. So that's being, as the big update to help drive home, actionable agronomic advice and really boil down 30+ metrics into just a simple page and straightforward page to highlight deficient areas, just to make it easier to take this complex data, and for a grower agronomist who just wants to look, hit the action items, this page really boils that down effectively. The other update is our soil biosustainability score, which you'll notice, excuse me, in the top right here, we have the biosustainability score, this is essentially our closest thing to an overall score when it comes to soil biology. And with the complexity and variability of the microbiome, as Marie was mentioning earlier, it's very difficult to provide an overall rating because there are 30+ metrics in our report. So this biosustainability score, which is actually, many of you are familiar with our soil quality index. This is pretty essentially the same index, except here we're providing a number to provide higher resolution insights on this score. So this soil biosustainability or soil quality score, what we consider it to be is a functional biodiversity score. So typically biodiversity index, they tend to look at just the species composition, not necessarily what those species do functionally or how they interact. So our soil biosustainability score, for those who are unfamiliar, is based on microbial ecology, and it's also benchmarked or measured against different management programs that we've evaluated across agricultural systems. So with microbes that tend to be more functional and more ecologically synergistic, microbes that tend to cooperate and build symbiosis with each other, we tend to see a higher score in terms of soil biosustainability, whereas in intensively managed fields with high degrees of tillage, high salt index fertilizers, lots of crop protection products, we tend to see a lower score with microbiomes where the soil microbes are falling into their own niches or maybe competing at excessive levels. So we see less of a cooperative soil food web, so to say. So that score, which was previously, it's still included in the soil quality section, but here we provide the actual number on the 0 to 100 index, giving this score in correspondence to our crop-specific database. So this would give you a good, overall in this particular corn sample, we see it's about close to the 50th percentile in terms of that soil biosustainability score. So the real, as Marie went over, the real focus of these updates has been on making the data that we provide more actionable and more simplistic to carry over into agronomic recommendations and management practices. So we're just going to go over 2 quick example reports, just this first summary page. And again, I should reiterate that the rest of the report will still remain the same. So all the 30+ indices across the soil quality, plant health, and nutrition categories will all be included as they typically are. This is just an additional cover page. So this is an example from what we would consider to be a poor report, and of course that's the definition of poor, optimal, or maybe a really good report varies by location, region, cropping system. But in this case, we're seeing a report that had a lot of problematic areas, and we see a little bit of a lower soil biosustainability score there at 34, so about the lower third of our database in terms of our soil samples. So charcoal rot here, anytime we see a disease that's in the very high range, that means it's really in the upper echelon of our database in terms of that pathogens abundance and in terms of the microbiome's ability to deal with pathogens. So looking at the microbiome holistically, we'd consider charcoal rot to be a high risk factor based on its abundance. So that's really, being one of those foundational factors in agronomy. We don't, when there's a very high level of pathogens, that they don't always have a significant impact on yield, but the likelihood is higher if we're detecting them at very high abundance in the soil, and we don't have the beneficial microbes to help keep them in check. So in this case, we saw charcoal rot show up as one of these major recommendations to improve and address. And of course, as crop consultants, agronomists, or growers, the logical solutions you might take away from seeing a disease pop out on a report like this, especially if it's a disease that's problematic in your region or crop, would be to, of course, look into disease-resistant seed varieties if possible for the particular pathogen that was detected. Biocontrol products such as biofungicides being another option that may serve depending on what's available, in your portfolio or in your region, or of course crop protection products, fungicides and others that may help suppress that pathogen and get them under control. And then in terms of nitrogen and potassium, our nutrient cycling functions, of course, this is where we look more so at biofertilizer type products, biofertility products, or other products that are known to improve fertilizer use efficiency. Because again, in the case of very low and low nitrogen and potassium metrics, what we're seeing here is not necessarily a lack of nitrogen and potassium, but rather based on the microbiome in these samples, we're not seeing very very high levels of nitrogen fixers, nitrogen mineralizers, the beneficial microbes that help unlock and make nitrogen plant available. Similar with potassium, we're not seeing many of those microbes that help unlock and efficiently make potassium available. So if the first page pops out like this on a report with low macronutrient functions, of course, what we'd look into product-wise would be potentially nitrogen fixing biofertilizers, so products that would introduce, say, azotobacter, azospirillum, or maybe rhizobium for legumes to help introduce the microbes that fit in this functional category. Or there's plenty of other products, including nitrogen stabilizers or urification inhibitors, that may help by ensuring that nitrogen is more bioavailable for plant uptake when the microbes aren't present, to help do so. And other products, of course, general biostimulants like humic acids that serve as natural chelating agents may also help in this regard. For potassium, we'd look a little bit, into products that, of course, make claims more so for potassium than nitrogen. So potassium solubilizing products, typically the main biological product class that would be able to potentially help out there. But again, other biofertility products like humic acids may also serve the purpose of improving nutrient use efficiency. So this is just intended to really help kind of hit those highlights, isolate solutions without having to go through the bulk of the report and compare, the different sections across the 30+ indexes that we have. And then for a good example of this is what we'd call to be, a more optimal or good report. We see a higher soil biosustainability score up at 75. And in this particular report, we won't go through it at length, but the macronutrient metrics and the disease risk metrics all looked pretty good. We saw, high levels of nutrient cyclers and relatively low levels of disease risk. So in this case, this is a report that, there weren't a lot of major areas of interest in, or major areas of concern in those those factors that are our primary drivers of yield. So instead, our algorithm, when it doesn't pick up red flags in those macronutrient disease categories, we start to look a little bit more at other areas that may be deficient and could use help with, because we know, with every field, there's not ever any perfect soil out there. It's always dynamic. The needs of the soil microbes as well as the plant are always changing across rotation, across season. So for soils that look pretty optimal, we can still find areas to potentially improve. That algorithm just looks a little bit further down the totem pole, or in this case, might look at functions that would be higher up on that plant health pyramid, beyond optimal photosynthesis and protein synthesis. But now we're getting to the microbes that help provide some of the secondary metabolites, like these stress adaptation promoters and phytohormones like ACC deaminase. So we see fungicide agents popped up because, again, if even though there aren't any fungal pathogens that are at very high risk, we like to see high fungicide agents to ensure disease suppressive soils. So a biofungicide product, again, being something that may help increase this category. Or if a fungal pathogen does pop up, perhaps a fungicidal agent or a terpene type product, or as a potential resort crop protection products. And then for the stress management hormone production sections, where we see low levels of salt tolerance promoting microbes and low levels of ACC deaminase producers, this is where some of those general biostimulants that Marie mentioned may come in handy. We tend to see a lot of those products like humic acid based products, kelp, seaweed extracts, or other plant-based extracts tend to perform pretty well across this section, which includes a lot of your plant growth promoting bacteria and fungi that promote, with products, biostimulant products that make claims of reducing plant stress or increasing root growth. A lot of times these are the sort of categories that we see them perform well in. So that would be the logical solution for a report like this that may look pretty optimal, but still has some deficient areas, some potential bottlenecks to yield, which may help push the needle in the right direction, even if this field already looks pretty solid, or has a relatively solid yield. So now we're going to hop in, and we're going to switch gears a little and hop into trials. And with BeCrop Trials, as many of you are probably already familiar, so no need to go into a ton of detail here. But really with BeCrop Trials, rather than sets of individual BeCrop tests that may be used more as a diagnostic tool, BeCrop Trials are intended to look through a more statistically robust lens at the performance of products or programs. So in the case of BeCrop Trials, we're doing replicated sampling over time in comparing treated and typically control or check zones. This allows us to run statistical analysis that really teases out and isolates the effects of the product that we're evaluating. So these are larger scale projects that adhere to more of a standard university level crop trial. So the main focus is being to track product performance, look at be able to measure factors like product efficacy and retention in the soil, evaluate and improve products, see where products may have weaknesses or areas that can be improved upon in terms of their formulation, and also, of course, verifying product claims and efficacy, showing that biological products and any other ag input products, including synthetic products or PGRs, showing what they can do in terms of the soil microbiome across our 30+ indexes. So the BeCrop trial report, just the process is, again, very similar to your typical university level crop trial, taking baseline samples in multiple zones that will be subjected to different treatments, and then taking samples after the treatment at least one, but typically multiple time points. And then our statistical analysis compares how the microbiome changed in both the control zone as kind of our background baseline, and then the treated zone to see how the product really, moved the needle over time in comparison to the control zone, serving as sort of our ambient check. And then the results we provide, I mentioned the statistical analysis, which many of you may have already seen some of our BeCrop trial reports, but the reports are a little bit more in-depth and detailed than our individual BeCrop tests, since these are these are just some excerpts from a BeCrop trial report here. And what we provide on the trial report are the most notable and statistically significant differences between the treated and control fields. So we look at the percent difference between the treated and control field in each microbial functional metric. So a positive percentage indicates there was an increase in the levels of microbes that support the function compared to the control, whereas a negative percentage indicates that there was a decrease. And then the highlights that we see here for certain functions, certain diseases, anything highlighted or outlined in orange denotes a statistically significant change. So we're seeing a change that was very likely driven by the product application rather than random variability. So this report is a comprehensive comparative analysis to allow the effects of products or programs to be looked at on a deeper and more scientifically robust level. But of course, just like with anything related to soil biology, there's a lot of complexity involved in interpreting these reports and really boiling them down to the highlights. And that's where our BeCrop 3.5 updates come in. We've revamped the scoring system on the first page of our reports, of our BeCrop trial reports. And the goal of this update is to simplify that process of highlighting product strengths in the results, and also tailor the results to the microbial functions of interest to the particular product type or even management practice that we're evaluating. So with this update, the new scoring system shows the cover page, which is not new. We're simply revamping the previous trials cover page. It highlights metrics according to what the product type will be. So for a crop protection product, for example, we would look more heavily at disease and biocontrol agents, of course, because that's what crop protection products, or say biofungicides, biocontrol products are making claims about or showing significant effects on, whereas secondary focus would be more of our nutrient cycling metrics and some of our other plant health metrics. For a biostimulant or a biofertilizer product, of course, we would favor nutrient cycling metrics and others that biostimulants and biofertilizers tend to improve most often. So that's part of our update as far as the revamp scoring system. And then the other aspect of our scoring system is new summary, the new way that we formulate the summary on that first page. So previously, for those who have seen our trial reports before, we highlighted the metrics based solely on statistical significance, or primarily on statistical significance. So the 4 metrics highlighted on the first page were generally those that statistically we saw the most major difference in, or the lowest p-value. In other words, the highest likelihood that the products drove the changes. However, statistical significance, generally we may see a statistically significant change of 10% in one metric. We may see another metric that increased 10%, but it wasn't statistically significant, but maybe still worth noting if it's a metric of interest. So in our old report, we looked solely at statistical significance. Now, with our new trial summary page, we highlight metrics based on both statistical significance and the raw percent change in those microbial groups. And we'll explain that system in a minute. We also previously did not have our soil biosustainability or soil quality score on that first page, and now we have added the soil biosustainability score if there is a major difference driven by the product or program that we're evaluating. And then we also now have the ability to generate multi-location trial report summaries. So for clients who are looking at answering the question of why products may work effectively in some soils but not others, we can generate an overall report that kind of touches on the highlights across multi-location trials to look for areas where the products did tend to push the needle more consistently across locations. So this is just a quick look at the old and new trial page. Again, the previous trial page showed highlighted the metrics that showed the most statistically significant differences. So again, highlighted it or outlined in orange is where we're seeing statistical significance. Now we have a much more simple scoring system that also may lend itself more easily to adoption by marketing teams as far as communicating the claims and the data that shows product efficacy and performance. So we've switched to this star scoring system with up to 3 stars being assigned, and the more stars that are assigned to each metric, the more effective the product performed in terms of that metric, both in terms of statistical significance and in terms of just the overall percent difference in the treatment versus the control. So even 1 star is still showing an improvement, but 2 stars is showing either more higher percent improvement or statistical significance. 3 stars is where we're seeing both statistical significance and a very high percent increase. And then again, we have the soil biosustainability score on the bottom of this first page. So I know we're getting up on time and want to leave some time for questions, so I'll try to zoom through this. But this is just a couple slides to go into more detail on how the star scoring system is applied to these reports. So the way it works is 1 star is assigned if a product showed a 10% increase in a beneficial functional metric. So for example, in organic phosphorus solubilization, that might be the case that we see a 10% increase. And if we see 100% increase, so a fold change, or in other words, twice as much doubling of that level of that microbial functional metric, then a 2nd star is earned. So we might see 2 stars in the case of a metric that increased over 100%. But then if there's a statistically significant change, so a low p-value, and our statistical significance threshold shows that the change was very likely driven by the product, then an additional star can be assigned. So even and this is all independent. So that's why we have and or here, because if an increase is if a microbial metric it does not increase by quite 10%, but it does show statistical significance, a star would still be assigned to it. So just, this is really boiling it down into a simple system where we can say, the more stars, the more effective your product was at improving these metrics, and hopefully can provide a little bit more clarity to folks on the less technical side, like marketing. And just to go through some examples here, so that now on the bottom here, we're going to show excerpts from this exact report. So for inorganic phosphorus solubilization, this is the excerpt from that actual section in the report. And we see that metric, which unlocks, these are the microbes that unlock phosphorus, increased by only about 7.5%. But that if we see it outlined in orange, that indicates it was a statistically significant change. So that's why 1 star was assigned, because it met this threshold for statistical significance. Even though it wasn't quite a 10% difference, it still was statistically significant based on the spread and variability of the data. And then with inorganic nitrogen release here, we see 2 stars. That's because here we see it increased by over 10%. It increased about 20%, 19.72. And again, highlighted in orange, it was statistically significant. So that's why the 2nd star was assigned, but it didn't increase 100%. So no 3rd star. And then when we look at salt tolerance, the plant growth promoter section performed great on this report. Those are hormone and stress adapter metabolites. That's our overall rating. But salt tolerance also earned 3 stars, and that's because it increased over 100%, increased actually 500%. So very, numerically a very high increase in this category. So that achieved the 2 stars, and then it also had a statistically significant increase. So that's what earned it the 3 stars overall. So I'll pass it to Marie. I know we wanted to save at least a few minutes for questions. So Marie, I'll pass it to you to go through some of our work on the horizon before we jump into some of our questions.
Awesome. Yeah, so taking all this information, I don't want to spend too much time here because there are a couple good questions, but when we look at trials across multiple locations, what we can start to do is really, really neat in creating these new product intel reports. So what these are looking at is comparing the product in the trial against our database of all of our products and benchmarking it against all of the products in various indexes, and then also looking at regionally where products will perform based on our database, where products will perform most effectively. This can help inform marketing and sales insights, help develop marketing material for products, and just add third-party validation, benchmarking your product against all products in our database. So what this requires is multiple locations, multiple soil types within the same cropping system. And we use it again to benchmark against our entire sample database and create these product intel reports. So this is on the horizon, and just wanted to, for those who are interested in incorporating this into trials in 2024, happy to discuss with anybody further. So that's what's on the horizon. It's really exciting, using multi-location analysis to benchmark biological products and really help differentiate products in the marketplace.
Yeah, so this is something we're piloting that's really exciting because for decades we've had these large scale models and tools for chemical fertility to look at yield curves and prescribe, fertilizer. We're now, and it's a great time for this because now with the advancements in AI and machine learning, we now have the ability to make sense of just such a large, our large expansive database of samples to potentially tailor and stack up products in terms of their performance, not only overall but across geography. So that's really exciting advancement in terms of what we can do, and ultimately delivering our goal of providing more actionable insights and building programs while taking the human bias out of it, thanks to the data-driven analytics. So now
Awesome.
I'm happy to hop into questions.
Yeah, there's a couple of good ones. I've answered a few just in text here, but there's a couple of good ones I thought would be good to go over with the whole group. One of the questions was, let me find it, there were a lot of questions here. So although a disease or pathogen may be measured in very high counts in a test, overall fungal biodiversity will dictate whether a pathogen becomes antagonistic or displays actual, symptoms in the field. Oh, sorry, it's hard to read all of these here. How does our test determine overall pathogen risk? What else is considered in going into that beyond just looking at pathogen counts?
That's a really good question. So we do look beyond pathogen counts. We don't just linearly look at the abundance of the pathogen and translate that. That risk level also does take into account the microbial ecology of the system. And what we have really the main way of looking at that, and what we integrate into it in its simplest form, is our resilience indicator. Our resilience indicator looks across the entire microbiome at its ecological synergy. And if there's, say, more healthy relationships between the microbes, like more fungicide agents that we see serving, versatile roles that help suppress fungal pathogens, if that resilience rating with, associated with the higher levels of, microbial synergy like fungicide agents is higher, that does curtail or decrease the pathogen risk. I'll, just for the sake of time, I'll stick to that sort of that's kind of more of a basic interpretation of how we look at it, but we do look beyond just the abundance of the pathogen and consider how the overall microbiome looks when generating those risk indexes. Great question.
And I would say, just to note here, this is what separates shorthand shotgun sequencing from the type of sequencing that we do. Is because we can look at the entire ecosystem and the whole function of the full ecosystem. We're not just looking at a linear disease risk based on this pathogen is here at this abundance, so you have high risk, which is how I understand other technologies. We're looking at this pathogen has this much abundance, and we also have these beneficials that are present, and we have these other things that are present that reduce the risk even if the abundance is high. So we take into consideration the whole disease triangle in our evaluation of disease risk. So I think that that's important to note. The other question that I thought would be good to go over altogether was around carbon. Do we, how do we look at soil carbon? Do we have an index that, might help understand soil carbon sequestration potential? I can chat about this one, Gus, but so the question exactly was, how are you collecting data to be able to measure improvements in carbon sequestration? So one of the index sections in our report is on the carbon cycle overall. So we're looking at things like organic matter release, carbon fixation. We're studying the whole carbon cycle. So the way that I think about this is we're measuring the changes in potential to sequester carbon. We're not measuring carbon itself. We're measuring the carbon pathways biologically. So what is your potential to sequester carbon? So that's how I would answer that question. Anything to add there, Gus?
No, that sums it up nicely. So we of course look at both. We look at the microbial functions that are involved in releasing carbon from the soil or net carbon loss. So these are your metabolic processes like fermentation, methanogenesis, and even aerobic respiration. And then we also look at the microbial pathways or functions that cause net gain of carbon in the soil, your decomposition or organic matter release and carbon fixation. So yeah, the overall carbon rating that we provide looks at the microbial potential to sequester carbon. The more microbes we have performing net carbon gain functions, the more likely we are to be sequestering carbon. Whereas if we have very high levels of microbes that are releasing carbon in gaseous forms like methane and CO2 from the soil, we see it lower. So yeah, we look really not just at carbon itself, but at the microbial dynamics of carbon.
Yeah, thanks, Gus. I think we're just about at time here. Maybe we have time for one more question. A lot of you asked about soil sampling protocol. We can email that to you directly. Let's see. Okay, this is a good one. Knowing the data is very helpful, albeit how do we use this data to take corrective action? So that's a good question. What we try to do is help our clients identify what are the most pressing issues and guide towards what types of products generally will help address those issues. It's really we like to leave it in the hands of the agronomists and the growers to find what product in their region actually works best and is available to them. So to say you have low salicylic acid, and we'll do a Q1 next iteration of our 4.0, but so I don't want to give too much away, but we want to give you directional suggestions on what index are the most hurting and where can you select a product in your market, in your area that's going to help address that issue. So there is a certain amount of autonomy here that's still left to the agronomist and the grower to make that decision. And we don't sell product. We are a partner to manufacturing companies, but we don't sell any products, and we try to remain a neutral third party on this specific product front. So what we really want to provide is, the diagnosis. What are the issues that are going to be the most impactful to yield and ROI, so that your agronomist and your growers, can guide to addressing those issues and position a solution to address those problems. I hope that answers your question. I think that's everything, and we're out of time here. If there are questions that we didn't get to, we'll send you an email with a response to those. There were actually a lot of questions here. Sorry we didn't get to all of them, but it was great to have you here. We love the participation in our webinars. They're super fun, and awesome to have so many engaging questions. You will get, if you registered for this webinar, you will get an email with the recording so you can revisit anything. And we can also follow up with those of you that asked for the BeCrop guide or our soil sampling protocol. So thanks everyone for being here. We'll follow up with anybody that's registered with questions and check in to see if you have any remaining questions about Biome Makers and the BeCrop technology.
Yeah, thanks for joining, everyone. Have a great day.